Hi, beautiful people. How are you? I hope you're having a great day. We're finally getting some really warm weather up in the 70s, and it's touched up into the 80s. We're still in Indiana, likely to get a few cold days, but summer's almost here. Today is part 2 of the 2-part series on the new research by Dr. Younger and his colleagues. We're going to discuss research on microglia and fractalkine as it relates to fibromyalgia and ME/CFS, and we'll sum up what the researchers found. Here we go. First, I looked at what previous researchers have found about fibromyalgia and fractalkine. They stated in their studies that fibromyalgia has less fractalkine in their blood. There isn't a way to determine how much fractalkine is in the brain yet, so they have to do the best they can, but it does give us an idea of how much fractalkine is in the body. And then I looked at a study by Younger and team published in July of 2023. This study was designed to determine if fibromyalgia involves an abnormal central response to a minor immune challenge. Meaning, if they put an endotoxin in the body, how will the body respond? The endotoxin was used to stimulate the immune system in these women with fibromyalgia, and we'll talk more about that in a moment. They used magnetic resonance spectroscopy, which showed an increased brain temperature in the right thalamus of the brain. And in the fibro group, 36.5 degrees Celsius pre-endotoxin to 37.09 degrees Celsius post-endotoxin was found, while temperature remained the same in the control group. That's interesting. It appears that the healthy control group was able to rid the body of the toxin, but the fibromyalgia group was not able to, at least not quick enough. Under normal conditions, the brain temperature is approximately 0.5 to 1 Celsius, or approximately 0.9 to 1.8 degree Fahrenheit, higher than core body temperature. Under neuroinflammation conditions, what's believed to be occurring in fibromyalgia, the brain temperature is 1 to 2 Celsius, or approximately 1.8 to 3.6 degrees Fahrenheit higher than the core body temperature. That equates to 2 to 3 degree Fahrenheit higher than what it should be. Also, in that study, researchers found N-acetyl aspartate, abbreviated as NAA in the literature, was decreased in the left relandic operculum in the brain. Numerous neuropathological conditions show this decrease, including brain injury, stroke, and Alzheimer's disease. So finding this in people with fibromyalgia is significant. This molecule in the brain is the most concentrated after the amino acid glutamate. It's found in neurons, oligodendrocytes, and myelin, and is synthesized in the mitochondria. It's pretty interesting. Basically, it's found on myelinated neurons. And that's why researchers who study MS, multiple sclerosis, find this important. But NAA is still under investigation to fully understand its purpose. However, this can be a sign of neuroinflammation or neurodegeneration. The endotoxin used in this study was lipopolysaccharides. An endotoxin is a toxin that is present inside a bacterial cell membrane and is released when the cell disintegrates. It's sometimes responsible for the symptoms of a disease. It's a heat-stable lipopolysaccharide substance, the technical term for endotoxin, of a gram-negative bacterium. Gram-negative bacteria have a hard protective outer shell and are harder to kill. They're becoming highly resistant to antibiotics. So the results of that study show a low level of neuroinflammation in the brain of people with fibromyalgia. A larger study should be performed to determine if people with fibro have an abnormal central response to low-level immune challenges. There wasn't enough evidence to support that hypothesis. So that's what Younger and team was determined to do. In a study by Younger and team in November of 2023, and Dr. Younger did a video on this topic, which is, of course, why I decided to cover this. And also, I believe one of you have been watching Dr. Younger's videos as well. And I've linked the particular video that's covering this topic today in the description section below. The primary interest of researchers in this study was leptin and fractalkine, and the cytokines that were secreted. We're probably not going to cover much of that, but we do want to look at leptin and fractalkine. It was a pilot study, meaning the first study just to see what happens before they do a large study. This one had just, I believe, 8 people with fibro and 8 healthy controls. They tested whether fibromyalgia is associated with altered immune responses. And in this case, to toll-like receptor 4 or TLR4 activation. I'll talk about that in a moment too. The results of the study suggested that fibromyalgia may have an altered immune response to TLR4 activation. We learned in that previous study that when we get exposed to pathogens, our body should properly fix the exposures. An anti-inflammatory effect should occur. But in fibro, it appears that the microglia that are supposed to do their job aren't doing their job based on the fractalkine response. In this November 2023 study, the women with fibro also responded with lower fractalkine, as well as, there's a whole list, I'll put them here, of cytokines. Perhaps that'll be for another day. On top of that, there was a greater release of leptin in these, perhaps for another day. So a lot was gathered in that study. Historically, some studies have not shown a significantly altered inflammatory biomarker profile in fibromyalgia patients. Other studies have identified elevated pro-inflammatory cytokines in fibro. In particular, TNF-A, CXCL8, and IL-6. These findings do show an underlying inflammatory mechanism occurring in fibromyalgia. Yes, yes, yes. Here's a quote. Numerous factors have been shown to influence fibromyalgia symptoms, including psychological stress, physical exertion, weather changes, impaired sleep, and hormonal fluctuations. The intricate relationship between these triggers and symptoms involves a multifaceted interplay of diverse pathophysiological factors, including aberrant central nervous system processing, neurotransmitter alterations, dysregulated pain responses, and immune disturbances. Notably, toll-like receptor 4, TLR4, has emerged as a particularly critical component of the innate immune system in chronic pain, thereby warranting further investigation as a potential target for fibromyalgia, supporting the hypothesis of sensitized immune responses mediated by TLR4 in fibro are two main lines of research, unquote. So toll-like receptor 4 is a key activator of the innate immune system, which is the first line of defense against invading microorganisms and fights bacterial infections by triggering pro-inflammatory reactions to eradicate invading bacteria. They're important, huh? We talked about lipopolysaccharide administration in our last video, and that's what they did in this video too. They determined that their previous work demonstrated that low-dose naltrexone, a TLR4 antagonist, reduces fibromyalgia symptom severity, and TLR4 activation in fibromyalgia through LPS administration, that lipopolysaccharide, in previous research replicates fibromyalgia symptoms. So it causes a person to get what is quite similar to fibromyalgia symptoms, including increased somatic and visceral pain responses. Diminished motivation, reduced positive mood, induction of sickness behavior, and impaired cognitive performance, all indicative of fibromyalgia. Together, Younger and team theorize that these findings show fibro involves a pathophysiological sensitized immune response to minor immune insults, which might explain fibromyalgia flares and our response to certain stimuli. Fractalkine, as we learned in the previous video, reduced microglia activation, a necessary part of the inflammatory response that tells microglia activation that everything's okay. So it's time to simmer down. In fibromyalgia, lower fractalkine levels may not be adequate enough to tell microglia to stop making our body sick. Leptin levels were found to be heightened in this study. It has recently been reported that leptin levels play an important role in systemic immune response and sickness response. In particular, leptin has been shown to sensitize microglia and increase their secretion of pro-inflammatory factors. In addition, leptin levels have been previously reported to be elevated in fibromyalgia. In particular, this occurs after an immune trigger and may be a factor in pain and fatigue in fibromyalgia. Well, if we thought fibromyalgia was complicated before, we can certainly agree that it's indeed complicated. A heightened expression of pro-inflammatory cytokines in endotoxin-stimulated human whole blood and peripheral blood mononuclear cells was found. An altered temperature and metabolic response to endotoxin also demonstrated abnormal responses. Dr. Younger in that video didn't go over those cytokine levels they found in the study, but in comparison to the healthy group, it was determined that it's too soon to interpret the significance of those findings. But if replicated by another study, it may prove specific abnormalities in the fibroimmune response. So, bottom line, what can be done about this deficiency in fractalkine? Dr. Younger stated that the first thing could be to give patients fractalkine and see if they get better even at any length of time in the fibro and ME/CFS population. Currently, researchers haven't given fractalkine in vivo, that is, directly to humans, but he is pursuing this area of interest. So we're going to hear more about this topic. So, what do you think? I look forward to more work by Dr. Younger. I hope you learned something. I send you gentle hugs and support. Love you.